Hello everyone, welcome to part 2 video of plant physiology chapter photosynthesis in higher plants. I have already uploaded part 1 video, so those who want to watch that, I have provided the link in description box. So let's start this part. So first topic is pigments in photosynthesis. So the first pigment is the chlorophyll A. It is one of the major pigments of photosynthesis. Its color is bright or blue-green in the chromatogram. It acts as a reaction center and it is used to trap and convert the light into chemical energy. Next is chlorophyll B. Chlorophyll B is yellow-green in color. It is an accessory pigment and it is used to collect the energy and then pass this energy to chlorophyll A. The third pigment is xanthophyll. It is yellow in color. Fourth is carotenoids. These are yellow to yellow-orange in color. Now important point about pigments is that blue and red region of the action spectrum shows higher amount of photosynthesis. Also, chlorophyll A is considered as a chief pigment of photosynthesis. Next topic is the light reaction. So first, light reaction is also known as a photochemical phase. It involves the following phases. First, light absorption, then water splitting, then oxygen release, and finally the formation of a high energy ATP and NADPH. There are basically two photochemical light harvesting complexes. First is PS1 and second PS2. PS1 is called photosystem 1 and PS2 is called photosystem 2. Now these light harvesting complexes are made up of hundreds of pigments which are bound to protein. Each photosystem has all the pigments except the chlorophyll A pigment. The chlorophyll A pigment is the only pigment which forms the reaction center. In photosystem 1, the chlorophyll A has absorption peak at 700 nanometer, so it is called P700, and in PS2 this peak is at 680 nanometer, thus P680 we call it as. Next topic is the electron transport. Now what actually electron transport is the movement of the electron. So here it begins as first the electron is present in the photosystem 1. This electron gets excited to an electron acceptor which is called phaophytin. So this movement is an uphill movement. Then this electron reaches to PS1 through a downhill movement and it passes first through plastoquinone, then cytochrome B6F and then plastocyanin and finally to PS1. Now again this electron will get excited to an electron acceptor called FES protein and then downhill movement to form NADP plus which will reduce to NADPH plus H plus ions. So first it was from PS2 to electron acceptor, then it reaches to PS1 downhill movement, then again uphill movement to an electron acceptor, again through ferredoxin a downhill movement it produces NADP plus reducing NADPH to H plus ion. I hope the electron transport is clear to you. This is called Z scheme due to the shape it forms. So next topic is splitting of water. So why splitting of water is required? It is basically to replace all the electrons to the PS2. Which electrons? The electrons which were removed during the light reactions. So how that can be replaced? This is achieved by the electrons which are available due to the water splitting. Splitting of water is basically associated with PS2. Water splits into H plus, oxygen and electrons. This splitting of water creates oxygen. This oxygen is the net product of your photosynthesis. So splitting of water reaction is 2H2O getting converted into 4H plus ion plus O2 plus 4 electrons. So it is located in the inner side of the thylakoid membrane where this water splitting takes place. So that's the end of this part. I hope all the concepts were clear. Please watch part 1 also so that you can easily understand this chapter. Thanks for watching.